Is it?
Dr. Jagdish Singh Lore. I am working as a principal plant pathologist and working on mainly on rice diseases. So my topic on emerging disease of rice in India. So how we can go ahead. Next slide, please. Yeah, if we see the occurrence of the disease from till 1965 or 1945. So first is the brown leaf spot. Brown spot, you can say that is the one become as a Bengal famine in 1945. No, this disease is serious at that time. So later on in 65, the blast is then we have seen in 1970 onward. You can say the bacterial blight is one of the major disease that we have not control in any chemicals because this is the pathogen is systemic in nature. So if we see the 85, then sheath blight will come when the high yielding varieties are dominant. Then sheath rot, 7 disease, then 10 disease, Bakane. Bakane is the one of foot rot. We can say other language foot rot is in basmati rice in northern India. False smut is also emerging from 2000 onward. You audible?
Yes, sir. Yes, sir. Yes, sir. It is audible, sir.
Yeah. I think it's not still screen mode, no?
Screen mode.
Yeah, in full screen mode, I think.
Full screen mode.
Yeah. In 2015, we have one new disease that is called the DKIG now. Earlier, Erwinia rot, but DKIG we have reported in rice. And last year, we have in northern part, it's not in southern India, it's in northern part, we have a new disease, first reported by PAU is southern rice black streak dwarf virus. It is also a challenge for us. Last year, it's the first time reported in India. So we have discussed. Next, please. Yeah. So we will discuss just these important diseases like bacterial blight, sheath blight, false smut, neck blast, brown spot are emerging, and now again southern rice black streak dwarf virus. This is newly emerging. Next, please. So actually, bacterial blight and sheath blight is not, we can say, emerging because this is the earlier is the major diseases. But still, bacterial blight we have managed through the host plant resistance. One is the success story, I think, in the South Asia or Southeast Asia because a lot of genes we have. But in Punjab concern, if we see this 5 or 6 time epidemics were happened due to bacterial blight, and in general it caused the loss 20 to 30%, but in Punjab 1980 and 81 it caused the 70% yield losses in crescent phase. Crescent phase bacterial blight was come. It is come in two ways: crescent phase and leaf blight phase. But the crescent phase was very dangerous and it caused the 70% yield losses that was published. So the work has been started in the bacterial blight at Erie in 1970 onward. The first variety resistance to bacterial blight have been released in Southeast Asia IR20 that has XA4 genes, and then onwards many varieties have been released. So resistance and many races we have identified. Next, please. Next, please. So in Punjab, we have identified the 10 pathotypes. 10 pathotypes because 4 or 5 time epidemics, and we have earlier in the 1980 or 70 we have only one race. Then in first variety we have released in 1986, that is PR109, and that is show the second race we have identified. Then in 1992 3, then 4, then what happened, we have released the 3 mega variety PR114, 116 and 118, and that varieties has XA4 gene, high yielding, very good quality, highly acceptable by the farmers in northern part, even in Haryana, in Uttarakhand, in J&K. So, but these variety also break down by the, in 2000, onward by the race number 5, 6, and 7. So these races we have identified, and we have released the 2007 one variety PAU 201 that has XA13 gene. So this variety is also breakdown by the race number 8 in 2009 or 10. And then after that, because single gene is not effective, then we pyramiding the genes in 2013 onward. And then we develop the variety PR121, 122. So the three gene combinations is also very good work in, I think, in whole the country. So that genes we have deployed in our varieties, and these varieties covered the 70% area in the Punjab, and also grown in the adjoining states in northern part. Again, next please. So these are the three races which we identified in 2010 and 2010, 9, 10. So race number 8 PBX, so Punjab Jantumna Suraiji 8, we have into variety PAU 201, then variety PR 120. That show also the another race, and one race we have identified from the Pusa 1121 Basmati. That races are very, very virulent on the new genes. I mean, the race number 10 is virulent on XA38 gene. XA38 gene is identified our team at PAU from Oriza, Nevada in 2008. That time we have 7 races and resistance to this gene, all the 7 races, but later on it has been susceptible by the race number 10. So similarly, race number 9 is also break down the some other PR113 our varieties. Next, please. So this is the identification of races. We have 10 races based on the near isogenic lines, because the work of Butiablite is the very established at ED, and they have identified the different genes, put in the one background IR24, and this is the near isogenic line to base the identify of races. So we have at the time 10 races maintained, and we screen our material against the race number 7 that is most dominant in the state. Next, please. So we also this 10 pathotypes sequenced by whole genome sequence. So these some races we have seen the phenotypic and genotypic are the similar, otherwise not all. If you see race number 8 is very close to the Korean strain, which we have references, that is phenotypically same. XA5 is not breakdown that race, but XA13 is breakdown. Otherwise, other races show the XA5 is breakdown by the race number 5 and 7 is very close, and race number 9, 10 is very close. And similarly, in the genotyping, this is also the same group. Please, next. So this is the analysis how the population of the Xanthomonas is work in South Asia and Southeast Asia, even all the countries of South Asia as well as Southeast Asian. So what we observe, because 4 genes, XA4, 5, 13 and 21, these 4 genes are widely deployed in the rice varieties, as in southern Samba Mashuri is also improved by the XA13 plus 21, XA5 13 plus 21, and many other varieties have been improved for resistance to butiablite by these 4 or 3 genes or 2 genes. Now we have a Basmati varieties with XA13 plus 21. So these pyramiding gene of these 4 pyramiding, this is very good working. But this is the individual gene data which show the frequency by the pathogen. If we see XA5 in red color, it's not effective in South Asia. It's most of the pathogen population, 88% in India, 100% in Nepal, become virulent on XA5 gene. But when you go in Southeast Asia, in Korea, there is no any strain which become virulent to the XA5. In Philippines, only 5% population. In the Vietnam is very low population. So XA5 is become virulent in India, Bangladesh, or you can say South Asia, but it's very good in the a Southeast Asian country. Similarly, if you see XA13 is well work in South Asia, but XA13 is not work in the Southeast Asian country. So this is the population. Pathogen population is very different in the case of bacteria blight. Qualitative resistance is available. Non-genes are available. Till date, we have about 50 genes, 48 or 47 genes for resistance, R genes known for the bacteria blight resistance, and 2 gene PAU identified that was designated XA38 and XA45. Again, please, next. Next, please. So this is the effectiveness of the genes based on the our literature as well as the our experience. So if you see the XA5 and 7 is very good in Indonesia. We have visited Indonesia and see the cultivars in the Java fields. There are XA5. Those hybrids have a XA7 or XA5 or other genes. These 2 genes which show the resistance. In Indonesia also. In the Philippines, there are also XA5, 21, 23 is the new gene from China, or is a glabrema that is also effective. In case of India, there are XA23 very effective, XA33 that is identified by IIRR, Hyderabad. XA38 we have identified that is also very effective in India. So in different countries, this is show how many genes and what genes are effective in the respective countries. So we can make a strategy to develop or to deploy the genes in the respective countries. Those are effective in that country. Next, please. So this work we have screened our rhizorupipagon accessions and other wild rices where we have identified the genes. So 2000 accessions we have at PAU, which we collected from ERI, and that all are screened. And in the case of rhizorupipagon, it has a very good resistance. 600 accessions approximately we have 300 accessions, 600 total, and 338 accessions show the resistance to the our pathotypes. Next, please. So this is the new gene which we identify XA38, and another is XA45. XA45 we have deployed in the our new variety that has been released, PR127, that variety, and background of USA 44, that is the mega variety. So this variety has XA45 gene, and XA45 gene is show the resistance to all the races still prevalent in the country. We have a diverse combinations of the races also that is show resistance to all the races. Again, next please. So then we pyramiding the genes. Pyramiding the genes, 3 genes, XA5, 13, 21 in our mega variety PR106. That variety has been released in 1976. Very good variety, very good grain quality, but is susceptible to bacteria blight. So in 2001, we have put these 3 genes. And next, please. So this is the another variety which are breakdown PAU 201 by the new race. So then this variety has XA13 genes. So we pyramiding the XA38, 13, and 21. These 3 genes we already deployed in our new variety PR128 and 129, and that has been released. So this is in Basmati. We have a that is called the Pakistani Basmati 386. So because this variety has a low yield and plant height is more, but we have tried to put the and susceptible to bacteria blight. So we put the bacteria blight XA13 plus 21 2 genes in this variety, and also the height is low and yield is 3 and 4 quintal per acre is higher than the earlier variety, and this variety is very good, same cooking quality, that is Punjab Basmati 3. This is also very good grow in the Punjab. Next, please. So this is some another pyramiding of the genes in South Asia and in different countries. Next, please. Pyramiding in different varieties, different cultivars, and mainly the genes which are deployed, that is XA13, 21, XA 51321 is the most popular combination to improve our varieties against the bacterial blight resistance in India as well as in the Southeast Asian countries. Next, please. Next, please. Yeah, this is actually varieties which we have released at PAU and how it break because we have a lot of epidemics, 5 6 time epidemic, every 4 or 5 year epidemics, epiphytic years for the bacterial blight earlier from 1968 to onward up to 2000 approximately. So we have work started on bacterial blight in 1980 and first variety we have released 1986, that is PR109. So some of the varieties which are breakdown as XA PR114, 116 and 118 is the breakdown. Then the PAU201 we have released in 2007, that is breakdown by the race number 8. Then 120 breakdown by the 9 and 10 number races. So after that we have pyramiding the genes XA4 1321 in the variety which has been released 2013 onward. So then we have comfortable and now we have no much incidence from last you can say 10 or 15 years, not much incidence we have reported in the of the bacterial blight in the Punjab state as well as adjoining states where the our variety also perform well. So next, please. So this is the varieties has a resistance 3 genes or 2 gene combinations. Next, please. So because these varieties are covered approximately 70% area. So this is the success story to manage the bacterial blight through host plant resistance in the Punjab state. Because however we have some susceptible variety Pusa 44, that is also a good variety and mega variety, high yielding. Farmers are growing in approximately 15-20% area. But in between that, our resistance variety which has a 3 gene combination that become a barrier among the susceptible varieties. So the bacterial blight has been automatically, you can say, through the barrier with the resistance varieties that is grown by the farmer, fortunate, and then we have good control or we have very successful management of bacterial blight through host plant resistance to develop a resistance varieties from last 3-4 decades. Next, please. So this is variety popular in the other states as Uttarakhand, then Haryana and J&K in Western UP. Next, please. Next, please. Yeah. So another mega disease you can say or major disease of rice now it's number 1 is sheath blight. Sheath blight is increasing year after years. If you see in 1980, 2000, 20 years, the severity of the disease 16 to 25% in India. But 2000 onward, that is become a double, 26 to 50% average incidence or severity throughout the country. This is reported in production audit survey by IIRR. So if you see earlier, this is 25% and then or almost double 2000 onward. This is due to the some our genotypes or cultivars released or input use. Next, so we have screened 15,000 accessions germplasm lines against this disease under artificial inoculation conditions from last 10 to 15 years. It is screened more than 30,000 accessions germplasm land races wild rice wild accessions, but we have no complete resistance against this disease. There is a partial resistance, or you can say, because the pathogen is necrotroph in nature, so resistance is very rare. But even then, we have a moderate level of resistance, and those are slow progression of the disease. and that lines show not yield losses even. So because only genetic resistance is not available much, so we cannot control this disease through only the genetic resistance. We have some QTL identified at PAU in India, in I think Southeast Asian countries. Many QTLs have been identified against this pathogen. So some transgenic approaches also used for the control of this disease, use the chitinase gene. Then we have also tested the level of susceptibility among the rice cultivars, and we can measure that with the yield losses. So not single approach work for the sheath blight control. So we go for the integrated approaches. What are the integrated approaches we have discussed next? So in the some of the cultivars, TTAP already published against the TTAP, the some QTLs, Peking from China, TTAP from Vietnam, Jasmine 85 from Thailand. So different QTLs have been identified, but still it's not these QTL working well. It's depend on the pathogen side, weather or microenvironment. Somewhere we have reported the more than 7 score on TTAP at PAU, even at other parts, and Jasmine 85 also 7 score or 0 to 9 score. You can see this is in susceptible categories. But even then, this is the slow progression of the disease. So we have identified some donor out of the 15,000 accessions which we screened. So some of the lines show the score continue good 3 to 5 slow progression as compared to these T-type jasmine and taking. Next, please. So what we see, we have a good this donor PAU 22769 that is show the very less disease as compared to the susceptible PR 121 variety is resistant to bacterial blight but susceptible to sheath blight. So we have a measure, we have a identified and we have a calculate the yield losses. So yield losses is until the disease is not reached to the flag leaf, then there is very less yield losses. So yield, if the flag leaf infection is higher, then yield is lower. Otherwise, if we stop the disease moderate level, then we have a in good position and we can manage this disease. So this is work done by my student, my PhD student. So we make a cross with these lines with PR 121 and we have a very good phenotypic variations. So these are the population size. Some of the lines has a moderate level. Even then, we have a also not resistance still. Moderate level and slow progression of the disease in the some 71 lines. 2 lines has very good, but some others lines show the 5 score or more than 5. Next, please. And some QTLs we have identified. We developed the mapping populations and the QTLs are present on chromosome number 1, 3, 5, and 6. Next. So this is what the another approach for the manage this disease. So how is the spatial spread of the disease among the varieties? So we have used the Swarna, Samba Mashuri, Swarna Sub 1, Samba Mashuri Sub 1, as well as our local variety PR 114, highly susceptible, PR 121 also. So if we see that we calculated in the center of the plot, and then we have collected the data spread, spatial spread of the disease to the all the directions, northeast, southwest. And this was published paper. And if you see the Swarna is the highly susceptible variety to sheath blight in the country. So we also measure the plant traits, how much number of tiller angle, that is also very closely related to the sheath blight spread. So in the Samba Mashuri, this has a very low disease spread as compared to Swarna. That is completely correlate with the farmer fields data. So in the farmer field, Swarna is highly susceptible as compared to Samba Mashuri in the case of sheath blight. So spread in the sheath blight is very high in the Swarna because after we have collected the data 2 week, 4 week and 6 week. the Swarna is completely become infected with the sheath blight within 2 weeks after the inoculation, but Samba Mashuri become after the 6 week. In 2 weeks, there is very low disease in the Samba Mashuri. So this is the comparison between the variety to find out the level of susceptibility among the variety to the disease. Next, so then blast, as we know, blast is also a major disease. So it's many time epidemics and more than 100 are genes already identified against the blast. But now is neck blast is increasing. Next, please. Neck blast is in the basmati rice. It's a very important disease because farmers are basmati rice is export in our country. That is, I think we export it more than 27,000 crore rupees every year, I think. So this is the neck blast, and even though genes are work against the blast, that is not work against the neck blast. That is the stage difference. However, the pathogen is same, but at the neck blast stage, in the many varieties, those have a blast R genes, those show the susceptibility to the neck blast. So we have identified, we have first developed the screening technique with Paspalum grass, infected grass, which we put on the neck and the panicle emergence, and the data will be collected after the 2 weeks. Then we see the very good lesions for the neck blast against this Paspalum grass bit method. So next, please. And we also identified we have a germplasm, 2K germplasm from ERI. That is, we have identified out of 300 this germplasm, whole genome sequence of this germplasm already done. And we have a 5 accessions which we screened at Himachal Pradesh Krishi Vishwavidyalaya Malan, that is a hotspot for the neck blast, and PAU with the artificial inoculation conditions. So 2 sessions show the 0 score in the case of neck blast. However, no any genes out of 100 R genes work against the neck blast. So these new, maybe there are new genes are present, and these accessions which we are putting in our mapping populations now for the neck blast resistance in basmati rice. So this is the 2 accessions and show the very good resistance to neck blast. So then false smut is already as a increasing over the years. Earlier it's known as a rich man disease. And if the disease come, then the farmers are happy to say this is the bumper crop. But now it's increasing over the years, and it's disturb the quality of rice. So it is, you can see in the 1981 that very less, but if on 2000 onward, its severity is moderate to severe form in the rusty color that dots show the high severity, which are increasing over the years. That is, I think, emerging disease. This is neck blast and false smut and brown spots. So next, please. So this is the our one analysis. This is more prevalent in the hybrid rice particular. So we have a analyze hybrid versus inbred. So in the case of hybrid, there are infected panicles, spikelets, more than 8, as compared less in the inbred. So this is also the similarly, we have not much good resistance against disease, and mainly, next please, mainly the problem is to create this artificially this disease. So we have developed and standardized the screening technique for this disease. That is the boot injection methods. So we artificially isolation of this pathogen and conidia show the 2 ml or 1 ml to 2 ml inoculum we put in the at the boot stage, and we have got very good symptoms against this disease. Use the boot injection method. However, it's very laborious, but any other method is not much successful except this one. Next, please. So these are the donors which we have identified, and for the false mud particular, we have a we have recorded the days to flowering. This is very closely related to the days to flowering of the crop of the accession. So same days to flowering, we have a 0 score and we have a 7 score in the germplasm. That germplasm we have specially collected with one collaborative project. This is the 2K panel from ERI and PAU collaborative project. So these donors now we have identified after 4 or 5 years of the screening artificially as well as in the hotspot locations. So these donors which we are using now, we have published my student it one in the European Journal of Plant Pathology. So these donors we are now widely used in our breeding resistance program. Next, please. So this is the same. We have some QTL identified for the false smut in different chromosomes. This is the association mapping of the germplasm. Next, please. So now brown spot. Brown spot is also we have identified very not much resistance is available, but few of the accessions after 4 or 5 years of the screening we have identified, and that donors we are using now in our program. Next, please. Because brown spot is now become a uh, very emerging disease in the direct seeded rice, because this disease come in under stress conditions. When plants are stress conditions, then the lenticels are open and pathogen is involved, enter in that lenticels, and it's caused the more severe disease. In the direct seeded rice, it's become major problem in the future. Now the direct seeded rice is grown, is the continue in this year lot of projects on the direct seeded rice. One we have project with the ED now. This year we have involved in that project on the direct seeded rice. So these problems are become a brown spot in the neck in the direct seeded rice. So these some QTS which we identified for the brown spot resistance. Oh yeah, this is the new disease which is the southern rice black streak dwarf virus, which we identified last year in 2020. The some districts, for six or seven districts had infected this disease. So this is very huge and cry, and 24,000 hectares of rice affected by this new virus. This is just we have identified based on PCR, and this is the very close to the sequence is very close to black streak dwarf. Black streak dwarf is the earlier reported in the, so it's reported in China in 2008-9. It's severe form in the China, but how it transmitted still we are not understand. So with this disease, you see the stunting of the plant, but this is not stunt virus, which are already in the rice. But this is the leaves are shorter, dark, and shallow roots, which easily uprooted. So last year it's very severe in some of the districts. This new virus is transmitted by the not BPH, WBPH, white-back plant hopper. And incidence we have reported 2 to 20%. This is the persistent circulative double-strand RNA virus. So still we have key questions for this disease. The migration, how it migrate. This is not seed-borne. How the WBPH come China from India. The alternate host is also many for this vector. So there is weather vector relationship is also important. So this is still we are working on this, and we have just now recommended to control the vector to the farmer. If last year we have reported this vector in the nursery stage, that's why maybe the disease is spread in some districts where the weeds are there and early rainfall is there. One is the reason. So we have tried to how we manage this disease. Next, please. So if I conclude it, so lot of genes already for Vitier blight, pyramiding the gene. Now the sweet genes are identified and already put in the IR64 background, sweet genes. So in the sheath blight, there is need to accumulative minor affect QTS We need to cross resistance between resistance for control the sheath flights and other level of susceptibility to know the susceptibility genes for neck blast, high throughput screening technique and donors, then false smut screening technique, which is very important for this disease. So identify the good donors and black streak virus, which just we are newly disease and we see how we tackle it. Thank you very much. So if any query, any question, please ask.
Yes, sir. Thank you for your commentable lecture. Your group, your student, as well as your breeders, along with you, they are doing a really a very good work for the country, not only for Punjab, for the entire country. Your people are doing the very good work. Really, we are congratulating and appreciating you people. And
Thank you.
yes, after hearing your lectures, I hope our students would have learned many things from your lectures and from your you shared your knowledge to our students. So thank you very much once again, and I request our students to ask some few more questions.
Yes, yes, yes. Sure, sure.
Sir, good afternoon. Good evening, sir.
Yeah, good evening.
Sir, I have a doubt, sir. Why
Yeah,
complete resistance not available for sheath blight, sir?
yeah,
Is
very
there any?
good question. Very good, excellent. Because the pathogen is necrotroph in nature. You know necrotroph, it's a first secretion of the enzymes, then dead the cell and grow on the dead cell. So you know 250 plant species become susceptible to this pathogen. It's attack on 250 species, plant species. You can say the same pathogen on the maize, on the cotton, on the chilies, on you can say potato scurf. We have some area under rice and potato. We have some area under rice potato system. So Rhizoctonia solani also scurf disease caused on the potato. But that potato Rhizoctonia solani strain not attack on the rice. However, rice sheath blight pathogen is attack on the potato. This is still we cannot understand how it's we have a one mega project on this potato rice system how it's attacked, but it's has a wide host range and necrotroph in nature mainly. That's why it's grow on the weeds, grasses, and the resistance in germplasm because it's necrotroph in nature. That's why the resistance is very low. So we have need
Sir,
only the susceptible level. Yeah. Yes. Yes. Please. Yes.
sir, is there any work is going on this aspect, sir, to
Which
identify? Sir, in this aspect, any other research is going on, sir, to identify
again,
complete?
yeah. So which I already mentioned, the if we go for the accumulate the minor effect QTS as well as we go for the level of susceptibility of the genotypes, which is depend on the morphological trait of the plant, which we already have experiments and published it. Tiller angle, number of leaf per tiller, number of tiller per plant, and plant height. So many these morphological traits we can include in our breeding program as well as the identification of QTS as well as the yield losses. So these, if we include these all integrated approaches, then we can manage this disease very well. Otherwise, it's now become number one in China, number one in India even now, because the pathogen is very, you can say very wide host disease and necrotroph and very good. So we can only manage it to reduce the yield losses. We have also done the experiment and published on yield losses. When it inoculated on the boot stage, tillering stage and flowering stage. So how much the incidence we have created and how much the yield losses. If the disease has a 50% or moderate level, there is no yield losses. Yeah, we can stop in between, I mean up to moderate level, then we can success.
Very good information, sir. Very good information. We thought that even a mild infection can cause loss to the maximum extent. But after lecture, we can know that mild infection, it is not going to cause much loss. So anyway,
Yeah,
it's
yeah,
information for us. Thank you,
Yeah.
sir. Then any more questions? Then if there is no more questions, I again, on behalf of our university and on behalf of our college and on behalf of our students, I once again thank you, sir. Thank you
Thank
for
you
all
very
your
much.
support. Thank you,
Yeah,
sir.
yeah.
Sir,